Okay, good evening, ladies and gentlemen. I think we'll make a start. Just past 8 o'clock, and we have a good few in the room. So look, you're very, very welcome to this webinar, which has been organised by the Teagasc Horticultural Development Department. My name is Andy Welton, and I'm your host here this evening. But joining me as my co-host is Owen Sweetnam. Owen is our recently appointed vegetable specialist who's based in the Teagasc office in Ashtown, and Owen looks after growers in the industry in the eastern side of the country. So look, delighted you can join us, and we're very fortunate tonight to be joined by an expert on our subject matter, a world-renowned expert with over 50 odd years experience in the whole business of Clubroot in terms of research and development and advisory. So we're delighted, and thank you, Geoff Dixon, Professor Geoff Dixon, for joining us from the UK tonight. You know, Clubroot is an old disease. We've been hearing about it since for a long time, but it's not gone away, and if anything, it's very current, and it's more serious than ever before. And Geoff is going to bring us his expertise on his lifetime's work on it tonight. He's going to talk about the mitigation of the disease through integrated control with a range of measures that will allow probably growers to live with the disease. So we look forward to that. But look, before we hear from Geoff, I just want to draw your attention to a couple of housekeeping rules tonight. Ladies and gentlemen, everybody's on mute, so look, you can't be seen or heard, and you're getting familiar with this technology, so I'd ask you to perhaps engage with us through the question and answer tab, which is located at the bottom of your screen, and it gives you an opportunity to ask those burning questions or make those comments to our expert tonight, Geoff. So look, please don't hesitate. Don't hold on to them. Click that box and fill in those send in those questions and comments. And Owen and I will look at them at this and screen them and make sure that Geoff gets to answer them. We'll record the session as we've done before, and if anybody has to nip off, or if you've got to go away during the course of it, feel free to watch it back. It'll be put up on the Teagasc YouTube channel in a few days, and Owen will explain how to access that later on in the evening before we finish. The other thing is people will be able to get IASIS points through this meeting, and because it qualifies for that. So those of you who are interested in that, again, we'll put up a link where you can fill in your details, and we'll make sure that that's sent to the people and that you get credited with those IASIS points. So look, without any further ado, we're going to try and finish up around 9 o'clock or thereafter. And Geoff is going to talk for about 45 minutes, and that leaves a bit of time for those very important questions. It is your meeting, ladies and gentlemen, and we really would encourage you to engage by asking questions or making those comments. So look, very quickly onto our speaker, Professor Geoff Dixon I spent the afternoon trying to paraphrase this man's career to date, but look, just to say that he is an internationally renowned expert in the study of Clubroot and its causal pathogen Plasmodioptera brassicae, based on, as I said, over 50 years experience in research and advisory work. His interest and expertise with soil-borne diseases and Clubroot particularly started in Cambridge, where he was appointed the founder of as vegetable pathologist at the National Institute of Agricultural Botany, NIAB. He then moved to Scotland and developed research groups working on biology and control of Clubroot in the universities of Aberdeen and Strathclyde, and that was in association with the Scottish Colleges of Agriculture. That's now known as the Scottish Agricultural University College, SRUC, and within that organisation, Geoff was the director of the Scottish Horticultural Advisory Service for a period of time. His research work over the years would have resulted in a very detailed understanding of the relationships between soil pH, calcium, and boron on the life cycle of the Clubroot organism. At the same time, the work enabled the development of practically based systems of integrated control for the disease. In this work, Geoff has worked and continues working with growers and the wider agricultural industries worldwide. He is Emeritus Chairman of the International Clubroot Working Group. His activities now continue through his consultancy company, Green Gene International, from Dorset, where he lives. And he's also a visiting professor at the University of Reading. A lot of you will probably know him through his writings in the Vegetable Farmer, and he has recently published Garden Practices and Their Science, and he's currently updating another very important Bible in the world of brassicas, vegetable brassicas, and related crucifers. So no one better qualified to speak on this very important topic, Clubroot. But look, before we come to Geoff, Owen and myself want to run a little poll, and Owen is going to handle this just to perhaps tee up Geoff's presentation, which we'll hear in a little while. So Owen, if you could just detail that for a minute.
Thanks, Andy. The poll should be on everyone's screen now. You're probably all familiar with the way we run these as well, and we appreciate your participation in them. And it'll help Geoff to get a feel for the audience that we have in tonight. So there's 4 questions. The first question is, how concerning is Clubroot in brassica production for you? Not a concern, a minor concern, moderately concerning, very concerning, or you're not sure. Question 2, on average, what percentage yield loss do you feel Clubroot is responsible for? 0%, 0 to 10%, 10 to 20%, 20 to 35%, 35 to 50, or greater than 50. Question 3, apart from crop rotation, what is your primary method of control for Clubroot? And we have resistant varieties, Perco, lime, or practicing good sanitation. And then just to kind of give us an idea of who the audience is, we've asked you to identify, I am a grower farmer, tradesperson, academic researcher, government agency, advisor inspector, student, or other. Okay. Yep.
Okay. Good evening, ladies and gentlemen. It's a great pleasure to join you in Ireland this evening. I have worked in Dublin quite extensively in the 1990s as external examiner to UCD. So I have a fairly well working knowledge of Dublin, not much else around in the Republic of Ireland, but I also worked for some time in Northern Ireland with a peat manufacturer. So you'll understand that I know a little bit about the island of Ireland. I've had a long career, as Andy has indicated, and I would also draw your attention that I'm a member of the Vegetable Consultants Association in the UK. I'm very proud of that because I'm probably the only academic who's ever been invited to join that group. And you are invited. You don't join, get yourself the vein made available. You are actually invited in. And the people who are in that group are consultants to the supermarkets and growers and all sorts of industry bodies. So they're people who are highly respected within the industry. I did about a year ago a survey of growers as to what they thought the importance of Clubroot is, and I surveyed growers in Cornwall, Fife, Lincolnshire, Dublin, and Lancashire. The answers varied, but basically they were saying, as I got from Fife, the disease is an absolute nightmare, and in Lincolnshire, the grower was losing up to 70% of his crop. The grower I spoke to in Ireland said he got a massive problem. It's a major problem for growers in Lancashire. And throughout Europe, Canada, China, it is, certainly so far as the Chinese are concerned, it's public enemy number one so far as their oilseed rape crop and the Chinese cabbage crops are concerned. And China is now the biggest producer of oilseed rape, but none of it, they don't export, it's all for internal use. Why is it such a problem? Well, it's a soil-borne organism. You don't actually see the effects of this organism until the infection is very well advanced. The organism itself is very long-lived. It can remain viable in soil for a generation, longer than 30 years. And it can remain viable in soil in that period without a host. It's very easily spread. Transplants, machinery, animals, wind, and work in Canada showed that you can spread it on potato tubers. If you buy potato tubers from somewhere that's got clubroot, the chances are you may have spores carried on the tuber itself. It's an extremely well evolved organism. It's only exposed to the outside environment for a very short point in its life cycle. It's in the soil as very resistant. Spores, they produce a very small body which finds its way to a root hair, and once it gets inside that root hair, it's protected by the host. And we now know from extensive molecular biological work that once it's inside that host, it's very heavily reliant on the host's metabolism for its existence. So it parasitizes the host. There's been very limited research, certainly up until the last 15 years. Now there is a vast surge of research fired off by an outbreak of clubroot in Canada, in the oil rape prairie provinces, Alberta, Manitoba, and Saskatchewan. And the disease is really causing the Canadians a big problem. And the oilseed rape crop in Canada, the canola crop, is worth an enormous amount of money. There are no agrochemicals, certainly, that can be used in Europe. There are one or two which are used elsewhere, and some of them are quite effective, but they're not environmentally acceptable. There are very few resistance genes, and it's a complex problem for breeders trying to produce resistant varieties. And I'll talk a little bit about that later. And over the last few years, certainly the research coming from Canada has shown that it can mutate very quickly. This was something that was never suspected in the past. Clubroot, it's a soil-borne disease, it doesn't spread very quickly. Well, we know it now, it does spread quickly, and it also can produce new strains which evolve capable of overcoming resistance, and it can do that very quickly. It's getting worse. Why is it getting worse? Basically, that's an outcome of global climate change. Wetter, warmer soils. In the past, it was held back in the winter. As the soil temperature went down, clubroot became inactive, and it wasn't expressed until the following spring. Now, with warmer winters, warmer autumns, it carries on multiplying and invading root systems. Crop intensification and the lack of rotations. Your growers, you will well know that you are forced into intensification, and it's very difficult in some instances to get a decent length of rotation. So far as vegetable growers are concerned, oilseed rape is a problem because it's a very susceptible brassica, and it's capable of building up large amounts of spores in soil. So if you're renting land and you're following an oilseed rape crop, the chances are you're going to run into clubroot as a big problem. As I've said, the causal organism, Plasmodioptera brassicae, it's capable of forming a lot of variants, capable of eroding resistance genes. And I'll just add to that, that if you look in some of the textbooks, you'll find it's referred to as a fungus. Well, it most certainly is not a fungus. It belongs to a huge group of organisms called Protista, And this is one of the reasons why it's been difficult to carry out research. You've got weed hosts and a protected lifestyle. The combination of those two, plus its ability to remain in the soil for a long time without a host, add to the problem. And as I said, up until about the year 2000, millennium, there was very limited research or funding. Research agencies tended to class it as a bit of a graveyard for researchers, and you found funding to do research extremely difficult. That's now changed. The outbreak in Canada and China has opened up the dollars and the Chinese currency. There is huge amounts of research now going on into the organism. What the grower will see, first off, is wilting. You'll see a crop which all of a sudden starts to show wilting symptoms. You may have irrigated it. It may be wet time, but that crop starts to wilt. And the reason it's wilting is that the clubroot organism has got into the root system and started to destroy it. This is your enemy. These are the resting spores that you find in the soil. You can have 100 million of those spores in a gram of soil, and you won't even know they're there. It is minute. The maximum diameter is about 5 microns. That's a millionth of a millimeter. So they are terribly small things, but they are quite capable of causing an enormous amount of damage. What they do, as you can see from this slide, you can see around the edge of that root hair, you can see these little fellas here. They're called primary zoospores. They're the product of germination of those resting spores, and they're attracted to the root by exudates which come from the root itself. And what they do, you can see just here, they inject themselves through the root hair wall, which is a very thin, easily penetrated piece of tissue. And once they get inside the root hair, they start to multiply and produce plasmodia. And these plasmodia then produce more zoospores, which can migrate into the depth of the root system. Here you see them as they're developing and maturing. And the net result is what you see here. Vast, great clubs. It has been referred to as cabbage cancer, which is a reasonable way of expressing it, although it's not. It does have some similarities, but we won't delve into that. So, integrated clubroot control. There are approximately 5 ways of approaching this. Firstly, land planning and use, soil health and improving its productivity, pH management and nutritional management, and controlling contamination, and the use of resistant varieties and their management. Land planning, rotation is a very important aspect for clubroot. You need at least 1 in 5 or 1 in 4, 1 in 5 to significantly diminish the spore population. And only then will you have reduced it by half. That's the reduction figure that you get. So you'd only begin to diminish the thing very, very slowly. You can use suitable break crops, which are grasslays, cereals, sugar beet, maize, legumes, alliums. Alliums particularly are very helpful. The leek crop is a very good break crop to control clubroot, because the allium, the leek roots produce exudates which are antagonistic to the clubroot organism. So that is a valuable tool if you can produce leeks. Avoid land which has been used for oilseed rape and forage brassicas. If you are renting land, then in advance you need to know considerable amount about what that land has been used for in the past. And growers I've talked to now, if they're renting land on a regular basis, they will keep records of that land, and now they'll use drones to go over and see what crops are being grown and make sure that they have the correct information. So you do not grow a vegetable brassica on land which has very recently had oilseed rape, or forage brassicas. Forage rape is another good source of clubroot infestation. Groundkeeping rape is a potential means of long-term survival. It increases it increases land infestation. Spring rape was always a bigger risk compared to autumn sown rape. It was. Now, as I've said, with climate change, during the winter, the oilseed rape is still growing and the clubroot is still active in the soil. Soil temperatures above about 5 to 10 degrees C, the clubroot organism is still capable of moving and in penetrating. So there is much less reliance on using winter rape. It used to be said that the winter rape would produce its components of yield whilst the clubroot organism was quiescent. That no longer happens. If you're a vegetable grower, the use of bed systems, which increase soil aeration and drainage. The more aeration and drainage you can get into the soil, the better, because those little primary zoospores, they rely on soil moisture in order to move across the soil. They're very small, therefore they've got very limited energy reserves. They can only move about half a centimetre before they run out of energy. So they need to be close to the root hairs and to get in, and soil moisture is one means by which they can locomate. Monitor, as I said, monitoring rented land ahead of use and keeping farm records of the land that you use is very important. GPS is a good means of disease recording and keeping an eye on what is being grown on fields and as part of the farm business management system. Also avoid land which has been heavily trafficked. Heavily trafficked soil is compacted, the drainage is impaired, and it will be very heavily waterlogged. Growers I've talked to down in Cornwall carefully avoid land which has been used for the narcissus crop, because that crop is heavily trafficked when they're lifting and when they're picking. Soil sampling and assessments of inoculum content, lab testing are vitally important. There are now some very sophisticated ways of measuring clubroot content in soil. The Canadians have developed RNA and DNA tests which enable them to identify the amount of inoculum present in soil and also its viability. So there are testing systems available. They are widely used. I was involved some years back in a clubroot infestation in watercress in Florida, and we used the Canadian Alberta lab to measure the inoculum content in the soils of the beds that were used for watercress production. It worked very well, and eventually we managed to cure the situation, cure the problem. Soil health and productivity. If you increase soil fertility, you increase the beneficial microbe populations, and beneficial microbes are the enemies of clubroot. We know that if you encourage some of the beneficial microbes, some of the beneficial bacteria in the populations, they will parasitize the clubroot. They'll parasitize the spore, and they'll parasitize the swimming zoospore outside the host, and you diminish substantially the amount of clubroot inoculum in the soil. In order to encourage that soil fertility, add manure and composts, but do beware of municipal composts, which can easily contain waste from home gardeners. They've decided they've got clubroot on their cabbages, so they chuck it in the bag and send it down to the local dump. The local dump turns that into municipal compost, and I can assure you that the systems of pasteurization, if you can call it that, used by municipal organizations do not kill off clubroot spores. Aerobic composting is not a reliable way of destroying clubroot spores. You can destroy it with anaerobic digestion. Deep tillage, breaking up soil pans, encouraging drainage so that the spores, if they're in the soil, are washed deeper into the subsoil below the rooting depth of the brassica crops, and aeration itself encourages vigorous root systems, which are capable of outgrowing the clubroot infestation. If you have sloping land, contour ploughing and cultivation and planting is a very effective means of preventing water flowing across the land, and the water can spread the infections across the land and down the land. So if you have sloping land, then practice contour ploughing and cultivations. Now we come to the age-old recipe for clubroot control, which was lime and grass. Yes, it's a good idea, but when I looked into this in detail many years back, the information was extremely confused. A lot of trials work had been done, asking how much to apply, when to apply, what to apply, but it was very little of it was actually quantitative and qualitatively analysed. I will show you in a minute some of the research that helped to reveal what is needed. Regular liming is effective. If you can raise the pH to 6.8 to 7, that will certainly help to reduce the infection that you get from clubroot. It is not foolproof. And we know now, certainly in certain parts of England where they've encouraged sheep down from pastures where the clubroot is present, they've raised their inoculum levels, and we're finding clubroot on soils of pH 7.5 and higher. I am very concerned that what we're looking at in some areas are clubroot strains which are competent to overcome liming. They will exist at higher pHs. What liming does is to improve soil health. It encourages the beneficial bacteria, which are antagonistic to clubroot, and the calcium itself is an important component of cell wall structure and strengthening. As a general rule of thumb, 1 ton of ground limestone per hectare will raise the pH by a half unit, but it does take time, anywhere between 3 to 6 months, depending on soil temperature and soil moisture. So in the conventional cultivation systems, you would put the lime on in the autumn and it would be having its effect in the spring. If you want to rapidly adjust pH, there are active limes available. Sugar beet lime is also very helpful. Sugar beet lime was used extensively in California at one time to cure an outbreak in their Calabrese crops. They had huge piles of sugar beet lime and they applied it to the land in California, and that quickly managed to bring bring it under control. There are now a clubroot outbreak in oilseed rape in North Dakota, and they are practicing the same process. They've got sugar beet lime there, which they are using. essentially what you're using is very finely very fine particles of calcium oxide, and they are very effective. Magnesium limestone will help to ward off clubroot and it also helps to control micronutrient deficiencies. Use boronated fertilizers. Boron plus non-ionic wetters are a very good prescription for drenching over transplants before you put them out. We found, as part of our research into calcium, that boron was actually as effective, if not more effective, in the control of clubroot than calcium. The problem with boron is it's very, very soluble, so it runs through the soil very quickly. You can only use it as a drench for transplants or something of that nature, and in boronated fertilizers as part of an integrated control system. Now, why is calcium so interesting? Why is lime so effective? This slide is one of many of a long period of research where we were looking to see what actually happens. And what you've got here are two levels of infection that we were using, 5 times 10 to the 3 spores and 5 times 10 to the 7. And as you increase the amount of calcium, as you see, the boxes fall away. The amount of infection in those histograms drops. What you're looking at here are different stages in the multiplication of the organism in the root hairs. And when you had low calcium, the organism was able to complete its complete life cycle within the root hair. As you increased the amount of calcium, so that proportion of multiplication dropped. Eventually, the organism stopped reproducing. It produced the primary stage, but it never went any further. So it didn't progress into the rest of the root system and didn't cause, and unless it gets into the deeper into the root system, it doesn't reproduce, and it doesn't produce the galls that you're looking for. We knew very well that there was a big difference in effect of concentration, and you can see it here. It was, you needed a lot more calcium before you began to get the effect on reducing infection. And you can see here that the organism, when there was a high concentration of the pathogen, was multiplying very heavily, very rapidly and very quickly, and going through its final stages, and was doing it up to 30 milligrams of calcium right the way through until we got to 75 ml per liter, ME per liter, milliequivalents per liter. It just justifies here why lime, but you have to bear in mind that when you've got high levels of infestation, even liming is only a very slowly acting action, only a very slowly acting way of mitigating. Now, it was mentioned in your survey about the use of Perco, and I'll come on to Perco in a moment. But increasing crop productivity by the following regular uses. Perco will encourage soil microbial populations which are antagonistic to the population. We know this is one of the routes by which it is active and which it causes, helps to overcome the problem. It slowly releases nutrients, nitrogen and calcium, which are in tune with crop root growth, and it's weather resistant. Unlike the ammonium fertilizers, it does not leach easily away into the soil. It remains in the root zone. It does not lead to nitrate pollution by leaching. The ammonium phase encourages root enzyme activity, increases the uptake of phosphate and potash, and it's synergistic with potassium phosphate. You can use Perco to encourage vigorous root formation and healthy plant and productivity growth. The calcium itself raises the pH and stimulates cell wall growth and strength, and discourages some foliar diseases as well. So you get an additional bonus. Where you've got early symptoms, as I showed you with that foliar wilting, the top dressing with Perco will stimulate growth, and you can encourage a root system which has been invaded with clubroot and is already beginning to show galls, and that will then stimulate the production of another set of roots, and the crop, you can get a decent crop as a result of that from a crop which previously was failing. You can link it with calcium nitrate as a top dressing, which again stimulates the rooting and gives you a good quality finish. So combinations of Perco, calcium nitrate, potassium phosphate, all of those will help in a managed program to control clubroot and allow a decent crop to be produced. Here you see on the right-hand side a root system which is coming from a plant which has been dressed with Perco. On the right, plants which hadn't been dressed with Perco. Well, on the left, rather, plants which had not been dressed with Perco. And you can see here how those plants that they had originally some clubroot in them, in the root system, how they managed to develop once the Perco had been added to a well-developed crop. Now, the contamination is an unappreciated problem in the control of clubroot. You can easily increase the inoculum content of soils by moving machinery around from land which is contaminated to land which is only moderately contaminated. This is why it's important to know about the amount of disease in the soil, because you need to know clubroot is not uniformly spread in a field. It occurs in hot spots, and you need to know how and where these hot spots are, and that enables you to avoid it. Clean machinery, moving from young crops to maturing crops, and from small areas of low infection to areas of high infection, rather than vice versa, helps to prevent the spread of the disease and the encouragement of increasing inoculum in the soil. Washing machinery also helps to avoid that disease spread. The Australians showed a good few years ago that irrigation water abstracted from watercourses, which is coming off contaminated fields, is a good way of spreading clubroot. So you need to know where your watercourses are coming from. If you're abstracting from a watercourse, you need to know where that water is coming from. If it's coming from land which is surrounded by oilseed rape crops, you are likely to be picking up contamination and bringing it onto your own farm. Surface floodwater and rising drains can again transfer clubroot between fields. This was why I was suggesting being careful about contour ploughing and containing the amount of drainage water going across land. It is easily windblown. This is something again which has come out of the Canadian work, because the prairies themselves are very windy places. These tiny spores can easily be picked up on soil particles and moved around holdings and from one holding to another. So wind blowing spores is a problem. And when I did the survey, it came out from the growers in Fife, which is again a windy area, that they reckoned that wind-blown spores was one of their problems. Potato tubers and ornamental bulbs will carry infection on soil which they are carrying. And so you need to know about, if you're growing potatoes as well as brassicas, and you're importing potatoes, you need to know where they're coming from and how clean they are. Sheep brought in from areas with clubroot will carry the spores on their hooves, their coats, and on their in their intestines, so their manure will be contaminated. And the clubroot spore is very resistant. It can pass through the gut, and the acids in the gut of most mammals is around about pH 8. They can get through that without too much trouble. As I've mentioned, municipal composts, if they're made aerobically, can carry the clubroot organism, and aerobic digestion helps to destroy it. If you are buying transplants from plant propagators or taking out contracts with plant propagators for transplants, you need to know something about the measures that they take for sterilization of the equipment and their surfaces, and the way in which they monitor it, and the sort of risk insurance procedures that they've got in order to control the organism and make sure that what you're receiving are disease-free plants. There's no possibility that you are importing, because if you import transplants with clubroot on their roots, then you're going to spread it across the holding, and once it's in for a very long time. Drenching those modules, transplants, with 15 parts per million sodium borate and a non-ionic wetter before transplanting is a good insurance policy in order to help control the disease. This just shows you the length that the Canadian growers are having to go to, or the people in Canada are having to go to. Alberta, which was where the infection was seen first, is the Canadians' oil and gas capital. The oil and gas companies want to run about across the land, and they are quite prepared to go to very extensive measures in order to clean off their equipment, and growers should do likewise. You can see on the left a very dirty piece of equipment, and the way in which the Canadians clean it up. They're cleaning it up on a bed, which underneath that bed are tanks that collect the water, and the water is then disposed of very safely. So finally, resistance management. Resistance is an extremely helpful tool for controlling any disease. It's probably the most economically effective means of controlling any disease, because once you've bought the seed, you've bought the resistance, and you've bought the capacity to overcome, or at least mitigate, the presence of the pathogen. The problem with clubroot is we've had resistant varieties on the market generally for about 10 years. Recent crops include the Brussels sprouts, cabbage and cauliflower from various seed houses. The difficulty is that the resistance genes are very limited in number, and the same genes are being used in the cultivars of vegetable brassicas as are being used in oilseed rape cultivars. So far as the pathogen is concerned, vegetable brassicas and oilseed rape are open season in terms of their variants which will overcome the resistance. In order to manage that resistance, you need to avoid land which has previously used oilseed rape, and especially rape which is resistant to clubroot. Because if you continuously bombard the pathogen with resistance, it will evolve tolerant pathogen races, and there is a huge number of variants which are available. Some are more damaging to vegetable brassicas than others. You should avoid cropping clubroot resistant vegetable brassicas in close sequence. So if you're using resistant cauliflowers, don't use resistant cabbage or Brussels sprouts, or likewise the other way around. As I've said, there's a significant shortage of clubroot resistance genes. There are 3 strong genes in Brassica rapa, which are the turnips and the oriental brassicas. There are a few polygenes, groups of resistant genes of very small effect in Brassica oleracea, which are the European brassicas, and there's virtually no resistance at all in Brassica napus, which is the oilseed rape and swede brassica. So all the breeders are fishing in the same pool to develop their resistant cultivars. There has been in Canada a huge movement to produce GM resistance, and it's been very successfully done. The Canadians are quite happily using GM resistance because they use GM resistance in their herbicide policy. What they found was that where they had GM resistance taken in, it lasted about 5 years. And so it was overcome. The tolerance was developing in Canada, and undoubtedly the Chinese are doing exactly the same, and the organism is evolving its tolerance very quickly. Finally, I'll just mention agrochemicals. We have had, over my lifetime, a huge number of chemicals that have come through with suggested ability to control clubroot. There was one in particular which was produced by the Japanese company Mitsui. It was very, very effective. And when you were running through trials, it was very difficult to maintain your objectivity, because as soon as you hit a plot which had been treated with what was called Nebogin, you knew immediately that's where you were. Unfortunately, so far as registration in Europe was concerned, it could not be registered because we could not find where it was going, and we couldn't be certain that it wasn't going to cause a considerable amount of time and problems in the environment, and partly because the Japanese had not tested it on avians and they didn't see why they should. So that's what resistance can do. You can see here on the left and on the right resistance lines, and a susceptible in the center. Similarly, here's a picture of 2 roots. Left side is resistant, right side is susceptible, and you can see where that red and white tape is. You've got a resistant variety on the right and a susceptible one on the left. It does work. It can be very effective. As I say, it's the cheapest way of controlling clubroot, but it needs management and it needs careful control. There we are, ladies and gentlemen. If you want to ask questions, I'll be delighted to try and answer them.
Great stuff. Thanks, Geoff. And as Geoff said, if you do have any questions, ladies and gentlemen, now's your chance. We've got the world expert. Now we're fine. We've got the world expert for the next 10 or 15 minutes, ladies and gentlemen. So look, there is a few questions coming in, and you can pose those questions through the Q&A tab down at the bottom of your screen. So please do. Don't hold on to them. Like I say, comments or questions are very, very welcome. Geoff, you did say you'd give us a very practical, broad talk, and that's what you've done. And like you've said, there's no quick fix. It's an integrated approach all around, and there's a lot of information there that people can take. I guess they'll want to go back over it and read it to really get into the detail, because you've mentioned an awful lot of good stuff there. So that will be available, as I say, when we put Geoff's.
Yeah, you're welcome.
Brilliant. The presentation and the recording will go up on our site in a few days' time, so you can go back over it again. But you mentioned chemistry there at the end. Yeah, I remember reading something a number of years ago about that product, that Japanese product, And I also read about products that they were using on potatoes like Sherlin and Ranman,
Yes.
Geoff, which I think were trialed back the years. Again, they never came to anything, even though I know they were never approved for use on brassicas, but they were never found to be any way useful, were they?
Some of them had effect, but they were never. There were environmental problems as soon as you hit the brassicas, and the registration authorities were never prepared, certainly as far as Europe was concerned, to accept their use, and basically quite rightly so. It would have been environmentally damaging to use them.
And the current range of fungicides that are being used on brassicas for control of diseases today, and you were saying you're updating, or you're currently writing an article for the Veg Farmer on that very topic. None of them have been trialed, have they, or there's nothing coming out of that?
I cannot conceive that there is anything. I cannot conceive that any of the agrochemical companies would go anywhere near even trying to test materials for clubroot control.
Okay.
But trying to produce a chemical which will control soil-borne diseases is so fraught with problems that I can't see them even beginning to put the money into it. It's a very expensive process, made even worse by the amount of information that you've got to produce for the registration authorities.
Of course.
So
okay.
The number of chemicals I've seen through my life is legion, and you'd think you'd got one that would work, and then lo and behold, 3 rats died in a lab in Iowa, and that was the end of it.
Okay.
That's my simple view.
Very good. Geoff, before we come to a few questions that have come in, most agronomists, and I'm sure I'm no exception, we've been called upon that situation that you mentioned, where you see those hot spots in the field where the plants are beginning to wilt, and you pull them up, and there you go, you see the finger and toe, as we call it, those typical symptoms. You alluded to the forms of lime there, and the use of the mixture of Perco with, I think you said, potassium phosphate. What's available here is Grand Lime a lot of the time to raise pH when things are in trouble. Is that form of lime, how useful is that compared to,
that's what I was talking about in terms of it's useful, but you've got to be prepared to accept that it will take 3 to 6 months before it actually produces the effect on the pH. So the pH does not rise until some good while after you've applied the material. Okay.
So that combination of, you mentioned Perco and potassium phosphate, you also mentioned boron. I mean, those kind of mixtures, would you recommend those when people are in trouble and they're
I'm sure that if you can make use of Perco and you can make use of potassium phosphate, and as I say, boron on transplants, all manage the infection with those elements. You have a tool which will allow, or a series of tools which will allow you to grow a decent crop.
Okay. Thank you very much. Owen, there's a few questions on the screen there. Do you want to just pull one or two, and we'll see what Geoff has to say?
One here, Geoff. What rate per hectare of Perco would you consider to be most effective, and what incorporation depth have you seen to have the best results?
Sorry?
What rate per hectare of Perco would you consider to be most effective, and what incorporation depth have you seen the best results from?
Normally you're applying about 400 per hectare and incorporating it into the top 10 centimetres, 5 to 10 centimetres of the soil.
Okay. Another one here, where are pathogen tests conducted?
I can add to that you can, if you incorporate it in rows rather than broadcast, you can get a very much more economic approach to using the material.
In and around the plants, where are pathogen tests conducted, and how many tests do you recommend per hectare? And how much roughly does a test cost,
Well, I'm not up to speed with how much the companies are charging these days, but basically as many tests as you can possibly do, but it's going to be expensive. And I've seen figures raised between 35 and 50 pounds a test. So it all depends on the depth to which these tests are done. If you are going to get a thorough PCR type test, then it's going to be quite expensive. You would want to test land in a W shape across a hectare, and you'd probably want 5 or a dozen test spots within that hectare. So it can be expensive, but it is very good information.
And with that, just on the tests, yeah, I've been looking at this in the last couple of days, given that we were having this meeting, and I mean, yeah, the tests can be anything up to £100 if you're sending them to the UK. Yeah.
If you're asking for molecular testing, the chemicals that are used are expensive.
Yeah.
I think probably all depends on the volume that you want done. I dare say some of the companies will reduce for a volume rate.
Yeah. But interpreting the results, Geoff, they're given, the tests will give you a spore calculation, won't they? The numbers of spores per gram of soil. And there's, it's kind of a traffic light system where, I don't know, less than whatever.
If you've got up to 1,000 spores per gram of soil, you are very unlikely to see symptoms.
Right.
Symptoms start to lift off between 1,000 and 10,000 spores per mil or gram, and thereafter it will escalate quite rapidly. So if you've got a low level of infestation, then that's the point at which you've got to start being extremely cautious about how you use that land in terms of spread from areas of high infection.
So how do you make that decision? It's if you have a medium, if you have 10 to 50,000 spores per gram.
If you've got areas with a medium infestation, you work from those to areas of high infection, and make sure that you carefully shepherd the low infested areas, because they're the areas where your liming, the Perco, and all the other elements will be very effective.
Okay, good. Another one here. Is there any value to fumigation with hot mustard? We tried this in the past, but it seemed to exacerbate the situation.
Biofumigation has been mentioned, and it's probably quite effective, but your problem with biofumigation is judging when you're going to incorporate that material and how easily it is to get it broken up and into the soil. The problem with biofumigation, as far as brassicas are concerned, is the biofumigation plants are brassicas and they're susceptible.
Right.
So, it's a devil and deep blue sea situation.
Yeah.
Where if you've got land which is relatively lowly infested, then I'd be very cautious.
Okay. Owen, I'll just take another one here and maybe you can come in again. Geoff, there's an attendee here saying they've read about a product called TTL Plus, which is used to overcome Clubroot in the UK. The article said it's a fulvic acid product applied as a foliar spray, and seemingly it encourages the Brassica to produce extra roots bypassing the Clubroot. Any experience?
It's a new one on me. I'd be interested to see the reference.
Okay, well look, we might get we'll get back to that attendee and we'll get a little, we can maybe you can talk directly to them on that particular one. Owen, we have a couple of more questions and we have a few minutes left. So is there any other one there that you want to pose to Geoff?
Yeah, we had another one here, Geoff. Which cultivated leafy brassica crop is the most susceptible to clubroot?
Calabrese.
Okay.
All right.
A straight answer there.
Yeah.
Calabrese is the most susceptible thing you can, or either Calabrese or Chinese cabbage.
Chinese cabbage was always the one I heard too. Yeah.
I've got some wonderful photographs of Chinese cabbage in Norway, absolutely murdered with clubroot.
Another one here, Geoff. Has there been any trials carried out with the added beneficial bacillus bacteria to the soil, along with a nutritional program to keep them alive?
Things like Serenade.
Serenade, yeah.
Again, Serenade has been suggested. I've not seen any quantified research that you could say proved its use. Serenade is a very valuable material, but basically it's used as a foliar spray.
Yeah.
And this would be my problem with this TT material that's just been mentioned. If it's been sprayed on, it's got to be translocated down into the root system. And that's not necessarily a very simple process.
Right, okay. Another one here on they're asking about the rate of that boron application to the transplants.
15 parts per million.
15 ppm, all right.
Sodium borate.
And is there any merit on the boron again? I mean, is there any merit on, applying that as a foliar in terms of the soluble to a field crop?
You would drench the modules. So you drench the actual peat that the plant is growing in. So you get it right into the root system. So it is then coating the roots and helping to prevent the movement of that primary zoospore into the root system itself.
Okay. Owen, have you any more there that you'd like to pose?
Just one here, Geoff. Are you aware of any treatment to irrigation water, for example, UV filtration, that will kill off clubroot spores?
Killing clubroot spores is a very difficult process. A lot of work was done at one time at Wellsbourne in trying to clean up stillages and transplant modules and things like that. It was found to be extremely difficult. Sterilizing water, I would think, again, would be a rather difficult process.
Okay. There's just another one there. Maybe we'll just take one or two more and we'll wrap it up. Soil biology was mentioned, Geoff. Are any of the companies investigating innovations around the use?
So I'm sure some people are looking at it,
right.
But I'm not sure that there's anything that's been published that's robust.
Okay, Owen, have you any final one there? Because it's going on for 9:10, and I don't want to be holding people unless there's anything else you've got coming through.
We can maybe, if there's any other ones, we can maybe ask if Geoff doesn't mind that we could just forward them on and just give a written response to any further ones. And the Q&A can be put up on the website, which I could explain now, Andy, if you like.
Yeah.
Where to find that?
That's fine.
Yeah, I think that's a good idea, Geoff. Look, we'll draw it to a close, I think, in terms of what you've been your contribution tonight, which has been absolutely illuminating, I think, and I hope our attendees found it.
I hope it's done some good, and as I say, if people have got questions, I'll try and answer them.
Brilliant. Well, look, we can arrange that. Some of the commentary coming in is very, very, very positive. But yeah, so look, before we wrap up, Owen, if you don't mind, we've had people asking how they can access, the recent webinars we've been having and the ones tonight's one. Can you just show people very quickly maybe how they can do that via the website? Yeah.
So I'll just share my screen there now. It should be up on your screen.
Yeah.
So if you put in Teagasc into Google, the first hit here is Teagasc, the Teagasc website. I'll just give you a very quick run through how to find where the webinar is.
In which case, gentlemen and ladies, I shall leave you.
Well, look, before you do go, Geoff, we were just going to give you a big round of applause, bula boss, as we say here. But look, can I, on behalf of our attendees tonight, thank you for your contribution. And like I say, we have found it absolutely illuminating. And hopefully we'll have you on again at some stage in the future. So thanks again.
It'd be a great pleasure.
Yeah.
Thanks very much, Geoff.
Thank you very much indeed. Have a good evening.
Good night, Geoff.
Sleep
So, yeah,
well.
will do. So before we finish up, ladies and gentlemen, as Owen was saying, Owen, if you can just share your screen again, it's just for the benefit of people. They've been inquiring how they can access the recent webinars and the information, and we just want to show you very quickly how to do that.
So this is the landing page of the Teagasc website, and you click on webinars over along the top here, and it'll give you a list of the Teagasc webinars through the sections, the different sections in the Teagasc departments. And if you scroll down, you'll find horticultural webinars here.
Brilliant.
Click on that, and then in here you've got all the webinars, the upcoming ones that are here that you can register for. And if you just scroll down through them, you'll find let's see, the soft fruit, vegetable webinar, like the carrot fly webinar that we had there in January, was it, or February? And you'll have the presentations and the videos available to view there, and the Q&A reports and the presentations of the speakers. So that's that, Andy. And I might just mention while we're on it, the upcoming webinar we have, we've on the Tuesday, the 11th of May at 8 p.m. We have Tom Robinson, who is an independent spray consultant from the UK, and Tom is going to talk to us about principles of spray application, how chemicals behave in plants, best sprayer setup, water volumes, nozzle choices for different targets, and he's going to as well mention the results of his application field trials. So we'll be sending the invitations out for that shortly, and that's on the 11th of May at 8 p.m. So I think that's it, Andy.
Good stuff. Thanks very much, Owen. So look, ladies and gentlemen, we won't delay you any longer. Again, thanks for joining. And if there was anybody there who wanted to access those IASIS points and you didn't get a chance to fill in the online in the chat function, just drop us an email and we'll make sure that it's directed to the people that look after that. So look, without any further ado, thanks again for joining, and we hope you gain something out of the hour's work, and we look forward to meeting you again. So good night, God bless, stay safe. Thank you.
All the best. Thank you.